So hi everyone, thank you for the introduction. I'm not gonna say more about myself because it was a very nice introduction already. So I'm gonna jump right into it. Already in the 1969, women took to the streets of Boston, demanding more research on their bodies. They even published a book. The book was named Women and Their Bodies. And if you just look at the contents table, because I went through the book, it was really interesting. There are all kinds of topics that came up that are actually still relevant today. But one sentence really stood out for me. And that was the sentence where they said, like, we emphasize that you take a mirror and examine yourself. And even after all, you are your body and you're not obscene, which is a strong statement, and it feels for me also a little bit as the cornerstone of citizen science and exploring topics that are not maybe mainstream in a way. Moving on, so for the last 40 years, there haven't been any major breakthroughs when it comes to bladder infections and vaginal infections, even though the WHO is reporting an increase of vaginal infections. Now, the vaginal microbiome, which is the topic of our research, is important for prevention of vaginal diseases, for successful fertilizations, as you said, and healthy pregnancies. It's not just for women, it's relevant for their partners, for their children. So a lot of people are doing research on the vaginal microbiome, but what we feel is important is that we also not just study disease, but also study what is healthy, what is a normal microbiome, to really define what is a vaginal healthy microbiome and understand the conditions. So a lot of research also within our lab has been always in collaboration with university hospitals for these logistics reasons. But we've noticed that when we have control groups together with when it comes to the vaginal microbiome, that these controls maybe not only represent the general vaginal microbiome of women. So we were wondering, like, is there maybe a lack of particular bacteria increasing in the Western world? What is the reason for this? Or maybe we just don't know what's normal outside the hospital. And that inspired us to set up a citizen science project. So our approach is indeed, as I said, citizen science, and that's the topic of this special event. We've published on this, so I invite you to have a look at our publications and look it up. But diving into it, so it has been mentioned already amazingly by the speakers before, citizen science can be an amazing tool to do research, to expand your research. We also want to advocate that it should be used in a conscious manner, in an ethical way. For those who know about the HeLa cells, from Henrietta Lacks, I would definitely advise you to look up the book, and there is also a documentary. But it is important that the research participants are not feeling like data collectors only. So there are all kinds of levels of citizen science on how you can participate. And with our Isala project, we try to function at each of these levels depending on the project. So I advise you to have a look at our recent publication where we go a bit more into detail what they each mean. Now, specifically for our project, Isala was named after Isala van Diest, the first female physician in Belgium. She was a role model. She was not allowed to study medicine, and she changed the law, actually, in short. Our project is just like any other project. It has lots of scientific goals. We're very ambitious. We want to study the healthy vaginal microbiome of women outside the hospital. We want to study what the impact is of lifestyle, environmental factors. We want to set up a biobank with beneficial bacteria, understand their mechanisms, but also see how they could be used as maybe life biotherapeutics. Now, what I think is unique about Isala is that we have also a whole set of societal goals. We want to break the taboo about intimate health, vaginal health. We want to empower women to not only understand and co-create science, but really to have a conversation with their friends about it and their partners, and bring science, women in science, more into the picture, as well as diversity. So that's, in general, a few of the goals of Isala. We try to do this with science, with communication, all kinds of tools. So as you can see here, this was actually one of our first sampling kits at the beginning of Isala, because we were looking actually for 200 participants to take a vaginal sample. At that time, we heard people saying like, people are not really going to want to do this. Women are not going to want to take a vaginal sample. It's kind of ew. So we thought, maybe it's just us thinking that it's so cool and nice. Let's put more effort into the science communication. Now, fast forward, this was 2020, so already 5 years ago. Within 10 days, more than 6,000 women registered, so that was massive. So this was published 2 years ago, but if you have a look, me see if I can get a laser. This is Belgium, where we started our participation. The top is actually the Flemish-speaking part of Belgium. And as you can see, we're very proud that we have participation all over the Flemish-speaking part, from the big cities that are a bit more heightened, but also from the smaller towns, which was our goal. So I advise you to have a look at the paper, but what I also want to mention is that our oldest participant was 98 years old, which is really inspired also further downstream projects that we have right now. So I'm going to go quickly over this, but about the results. But as you can see, our question was, how healthy are the vaginal microbiomes? Which bacteria can we find in there? And we found mostly that, as you can see here, that women were dominated by a range of lactobacilli, also diverse bacteria, but we could group them in general. And this is a very simplified plot. And I remember our computational biologists in the group, they hated pie charts, but people actually love pie charts. So this is more for science communication purposes. And we try to communicate in an easy way as well. Also from our questionnaires, microbiome analysis, it's always important to have a range on this lifestyle and all these environmental factors. These are also very interesting data to communicate. So we do that also. We select a few, not too much, and communicate them, and not just in short infographics, but we try to also write blog posts on so people can understand them in a broader context. We also, on our website, if you have a look on isala.be, we have also an English version where we communicate about these bacteria. For example, for Lactobacillus crispatus, which is one of the most dominant ones, we make a little bit of a passport of the bacteria, what it looks like, what it does, where you can find it. This helps participants also to understand their personal results, which they get. So we do personal results and we do general results, and we try to even communicate to medical professionals if they come across our page. So this is an example of one of my own results years ago. So we give them a pie chart, a table, what they can find back, and you can click further to better understand these bacteria. So no medical advice. Afterwards, we send out a feedback survey asking participants actually if they understood, and if it was clear. And on more than 2,000 participants, then actually almost 100% would participate back again in Isala. So this got a lot of media attention, which for us felt like there was an unmet societal need. So that was really nice also to get that feedback in that way. What I think is really nice about Isala is we went already out to conferences and just events and talked about our project without our paper already being published, because if you wait for a paper, we had to wait 3 years. And this inspired groups all over the world to do something similar. They said, I also want to study vaginal microbiome in my country. And since we've done all this effort in setting up this communication stream, a strategic way of communicating, we said, we definitely want to share the way we do it. And this is a lot of bottom-up initiatives. And as you said at the beginning, we have already sister country projects all over the world, almost in 20 countries. So they are also all named to their own role model. Like, for example, Laura is named to a role model in Peru, Fatima in Morocco, and so on. So these are, I think, pictures from in Cameroon with Josiane, the Lekki project. She also gave a presentation a while ago at MVIF about how she went to rural tribes to discuss the vaginal microbiome with the women. We have Nigeria and Peru as well. But I'm going to go much faster because I have a few more slides. So we published also a perspective on this global sisterhood that we have. Being a lot in the press, I would say, sometimes with Isala, we get a lot of questions like, what can I do on my microbiome? And as a scientist, it's very important to communicate to have integrity and to not immediately give clinical advice, which we cannot do, and be honest about the stage that we are. So scientific literacy is something that is very important within Isala. So we try to communicate about associations and causation, but we do say that, for example, there are all kinds of lifestyle and environmental factors that could have an impact on your microbiome, which is something that we study as well. And you can find it also in our manuscript. But this is, for example, something that you would never communicate to the broader public. So we try to do it a little bit more bite-sized in infographics. So I have a few examples. I'm going to go quickly through them because of the interest of time. So, for example, we saw a big impact of childbirth and pregnancy, so that we would communicate in this way. But afterwards, of course, we always say, like I will say it at the end, this is an association and not a causation. The same for hormones. Contraceptives were a hot topic still actually, but 2 years ago even more. So we always try to link and understand how this association between the vaginal microbiome is with contraceptives and menstrual and hormonal fluctuations during the menstruation as well. Sexual activity. And then we also have a few follow-up studies, actually. One of them is Jean Doe, trying to see if we could use the vaginal microbiome as a predictor of sexual intercourse in sexual assault cases. So these go a bit deeper into causational studies as well. The same for hygiene practices. We saw a link with menstrual pads and the vaginal microbiome. So now we have the Luna project where we have an intervention study studying all different types of menstrual hygiene products and the impact on the vaginal microbiome. The same for diet as well. So we try to communicate these aspects, but always mention that at this time we could only explain around 10% of variation with these lifestyle factors. For the gut, Jeroen Raas and colleagues have published around 7.6%. So we have to definitely put this a bit more in perspective, like how much weight we put to these terms. And that's always important in citizen science. And I think I want to quickly also say it's very important to always have a look at who's participating in your project and how inclusive you actually are and what the demographic profile is of your participants. So we did that, and we saw that we were missing some groups. This is an example of Moroccan and Turkish women, but we also have, for example, less women in poverty. And we have set a sociological study with focus groups and in-depth interviews to better understand why someone would want to participate or not participate. And this is a set of tips and tricks that we got about awareness, being more as a researcher to be aware of your diverse community, and to communicate not only from your own perspective and background, that you should consider also the language that you use, so not just translate, but the message that you're using. Trust, so not every, that was also mentioned by one of the speakers before, not every group of society trusts scientists with a reason of historical background. So you should always see who is a trust person of a particular group. Taboo, understand that some groups have some taboo on some topics, but sometimes we might be surprised by that some groups don't have the taboo that we think. And subtlety could always help in some topics as well. So with Isala, I just want to say that we've entered into a third phase already with 6 new projects where the findings that I showed you before, the associations and the links, we go a little bit deeper in better understanding the mechanisms, the causation, and sometimes you do this in a citizen science matter if it can be beneficial, but sometimes it's not, and it doesn't have to be. So one of the recent projects that we just finished is the Rufaida project, where we studied the impact of Ramadan fasting on the gut and the vaginal microbiome. So the samples are now being analyzed, so very excited. In this project, an example, for example, is iftar or eat and food. At the end of the day is very important. So we did like a small contest that people could send pictures of their eat table, and then they could get an Isala goodie bag. So it's a small example for of how you could interact in one way or another. So this is quite recent, so I thought of it. So and then I just want to end by saying that we studied this in the initial, what we wanted to study is how healthy are the vaginal microbiomes in the general public. We had lots of people participating in Belgium and now expanding all over the world, studying all these influencing factors and now going a bit more deeper into understanding why or why not they could be linked to the microbiome, isolating all kinds of bacteria. This is an older figure. It's now more than 3,000 bacterial isolates, vaginal bacteria that we're studying in the lab, going international. And still our goal is to develop better diagnostics and therapeutics to help women as well. And we've been very fortunate to be awarded for our science communication. And with that, I want to thank our whole team on working on this amazing topic, and our funders, but more specifically our Isala participants for being so open in participating and sharing actually their experiences. And that was a bit my talk. Thank you very much.